will briefly introduce our company, and then I will share my thought on VS101. We developed a strategy. Well, let me introduce VSPharmTech briefly. VSPharmTech was founded in November 2018, and is now a venture company with 7 years old. With the slogan "Discover the Better," we are now focused on finding better solutions for cancer treatment. Over the past 2 years, we have gained confidence in our anti-cancer research by receiving approval for 3 clinical trials from the US FDA. Furthermore, a new drug under development in phase 1 clinical trials demonstrate its potential by achieving 100% CR rate without DLT. We have built a strong network with the leading cancer research institutions worldwide. We conduct phase 2 clinical trials in the US, and we hold a leading market position in securing orphan drug designation for GBM. As a member of JLabs, we've connected with 11 pharmaceutical companies. The name of VSPharmTech was created by CEO Shin Park, who is staying over there. VSPT is an acronym for Vita Scientia Pharma Technology, a Latin phrase meaning life science pharmaceutical technology. Okay. Actually, I'm not an expert about the DDR. For the past 20 years, mostly I've been working in the CGT field in Korea and Japan. Check out the image below shows for cell therapy products approval while I was working at venture companies. Here's our VSPharmTech management team. I'm very proud of them. They're all experts who have worked in the pharmaceutical industry for over 20 years. So VSPharmTech has sufficient capabilities to conduct basic research, clinical trials, and regulatory affairs in-house. And then I'd like to explain how VSPharmTech developed VS101, a radiosensitized-based treatment for various cancer, including GBM. We have received technology from the Korea Institute of Radiological Medicine, called KIRAMS, for a composition that effectively kills cancer cells when used in combination with radiation. And we are developing into an anti-cancer effect enhancing agent that promotes cell death. Although this substance itself is essentially non-toxic to cancer cells, it has been shown to have a cytotoxic effect on cancer cells when combined with the radiation. We had to devise a strategy to develop this substance into an anti-cancer drug. This is because the drug repositioning strategy aims to develop treatment for specific disease by focusing on demonstrating efficacy in preclinical and clinical studies based on already available toxicity data. So we've identified 4 key points. First, the characteristics of drug. Second, understand the radiation therapy. Third, figure out which type of cancer to target. Fourth, understanding SOC. The active pharmaceutical ingredient API of VSPharmTech is an antipsychotic drug that is being repurposed as apoptosis enhancer. It has been suggested by many scientists that antipsychotic medications may enhance the effectiveness of radio and chemotherapy, and in vitro studies have been conducted to verify their intracellular effect. As a result, it was reported that various mechanisms and phenomena were discovered when antipsychotic drugs were used in combination with radiotherapy and chemotherapy. Ionizing radiation, IR, damages the DNA of living organisms in various ways. For example, it causes single-strand breaks, double-strand breaks, and base and sugar damage, DNA cross-link damage, and cluster damage, etc. Radiation therapy uses high-energy radiation and works through two mechanisms: directly damaging and DNA of cancer cells, and indirectly damaging them by generating free radicals. Cancer cells that have had their DNA damaged by radiation like this must decide whether to repair this damage. As well know, if the damage is repairable, the cells activate DNA repair pathways, and if the damage is deemed too severe to be repaired, it induces the apoptosis. You are probably already familiar with the basic fact that damaged DNA is repaired through the following pathways. Of these, the most severe double-stranded breaks is repaired through homologous recombination repair and then non-homologous end joining repair. As previously explained, ionizing radiation and anticancer drug activate various signals that directly and indirectly affect cell survival. Therefore, based on existing research data, we predicted the MOA of VS101 when administered in combination with radiation or anticancer drugs. Based on a discussion with the radiation oncologist, we selected 4 types of cancer. The cancer had to be a type 4, which there were no chemotherapy treatment options, or that was resistant to chemotherapy. And it had to be cancer where radiation therapy played a significant role, which was also consistent with the recent trend of organ preserving treatment. Among them, we choose head and cancer, which is easily accessible for animal efficacy tests and clinical trials. To confirm the in vivo effect, we conducted a mouse experiment to verify the effect on head and cancers. The mouse model was X-rayed and simultaneously administered VS101 orally for 3 days only. Treatment was discontinued, and the mice observed until tumor volume reached 1,500 cubic centimeters. Since this was our first experiment, I was very worried. However, the results when radiation therapy was combined with VS101 were quite surprising. The study results showed that radiation therapy was significantly more effective than the control group and drug therapy alone group. Compared to the groups that received a single radiotherapy, the tumor growth inhibition TGI effect increased by more than 60%, and the survival rate also increased by more than 30%. We repeated the same experiment several times and obtained a similar result. So, just curious about that. I couldn't help but wonder. So we decided to investigate the specific MOA based on pharmacological and radiological mechanisms. This is a schematic diagram showing initial MOA of VS101 in cancer cells based on results confirmed through research experiments at that time. Because this drug is being repurposed as an anti-cancer agent, numerous studies have investigated its effect on neuronal cells. But relatively few studies have examined its impact on cancer cells or its anti-cancer efficacy, and the experimental findings remain preliminary data. Through brainstorming, we developed several hypotheses about the expected mechanism of action, prioritized them, and tested each one sequentially. Therefore, in mouse experiments described above, we first examined the cleaved caspase-3 and cleaved POP, markers of apoptosis, and 4-HNE, a marker of ROS production using frozen cancer tissues. Like the tumor suppression effect, we observed a significant increase all three parameters when VS101 was combined with radiation compared to the effects of VS101 alone, radiation alone, or the control group. To obtain more definitive data, we decided to conduct using a breast cancer cell line that demonstrates significant efficacy in vitro. Because radiotherapy and chemotherapy are often used together in cancer treatment according to SOC, this study aimed to investigate the effect of combining VS101 with radiotherapy and cytotoxic drugs. VS101, IR, and we used doxorubicin were administered once a week for 3 weeks, and the effect was observed. The experimental results show that VS101, doxorubicin plus IR, the triple combination group showed the highest tumor suppression effect. Next, the doxorubicin plus IR combination and the VS101 plus IR combination groups showed similar effect. All combination groups showed higher effect than the IR single treatment. And then we stained the obtained cancer tissue to observe the DDR-related markers and apoptosis markers. Tissue results show that combined administration of radiation, VS101 and doxorubicin increased the expression of gamma H2AX and phospho-CHEK2, which are involved in DNA damage and repair response, and cleaved POP and cleaved caspase-3, which are involved in apoptosis, consistent with the established tumor suppressive effect. Therefore, we hypothesized the following. When DNA is damaged by strong radiation causing double-strand breaks, ATM, CHEK2, and gamma H2XR expressed to promote homologous recombination repair. But VS101 inhibits or regulates the expression of RAD51, thereby disrupting HR repair and inducing apoptosis. To confirm this hypothesis, we examined DDR markers in vitro in prostate cancer cell lines to determine whether they were also expressed in other cancer cell lines. Other DDR markers were also examined, but most remained unchanged. However, Radial-51, which is involved in HR repair, was continuously reduced by VS101. Western blot and foci analysis confirmed that the expression of gamma H2AX as well as that of phospho-ATM and phospho-CHEK2 increased over time following the combined treatment with VS101 and IR. Immunocytochemical analysis demonstrated that Radial-51 expression was significantly reduced when VS101 was administered and in combination with radiation. And it was also decreased when VS101 was given as a single treatment. And we simulated the docking potential of Radial-51 and VS101 through docking studies. The result confirmed that VS101 is highly likely to bind to pocket near domain 1 and domain 2 of Radial-51. We are continuously conducting various experiments to obtain more definitive research results. And in the meantime, we were preparing for IND approval for GBM last year. The 4 key points above, information of disease and association was required. Dr. Samuel, who is an expert in this field, explained the characteristics of GBM in great detail yesterday. Therefore, I will only mention it briefly. The median overall survival after treatment is known to be about 1 year. Why GBM treatment is difficult, there are 4 main reasons: invasiveness, and blood-brain barrier, and tumor heterogeneity, and treatment-resistant cancer stem cells. So there is a reason why chemotherapy alone is not a cure. Limitations of drug delivery. Most anti-cancer drugs cannot cross the blood-brain barrier. Intrinsic drug resistant GBM cells are either initially resistant to drugs or develop resistance rapidly. Slow division of cancer stem cells. Many anti-cancer drugs target rapidly dividing cells, but GBM stem cells divide slowly, making them less effective. The alkylating agent temozolomide, called TMG, is also frequently used in monotherapy or in combination with radiotherapy. But the side effects of TMG are generally mild. However, despite TMG being the primary chemotherapy agents for GBM, the problem is that approximately half of patients develop resistance to the treatment. That's the mechanism of temozolomide. The very important thing is MGMT. Temozolomide primarily adds mature groups to DNA bases. When the active MGMT is present, O6 MAG is repaired without cytotoxicity. But if MGMT is inactivated or fails to fully repair or O6 MAG, it ultimately leads to double-stranded breaks, resulting in cell death. And also, Furthermore, it is expected that simultaneous exposure to high energy radiation will further increase the rate of double-stranded breaks formation. Mentioned earlier, we hypothesized that if VS101 could downregulate Radi 51, it would be effective in treating GBM. Radi 51 prevents a basic site that occur during the BER process from leading to chromosome breakage when they encounter replication fork. Also, the ping model was created as in the previous experiment. Radiation, chemotherapy, and VS101 were administered, and luminescence intensity was measured. As in previous trials, the best results were achieved when IR, TMG, and VS101 were administered in combination. Interestingly, a significant improvement in therapeutic efficacy were also observed when TMG and VS101 were combined. Based on this, we received the approval for phase 2 clinical trials from US FDA. And this cartoon shows the familiar concept of synthetic lethality. So I will suggestion. Let's think about this more deeply. Does the problem necessarily have to arise only at the genetic level? And do only two factors necessarily have to be involved? What if, as in the example above, TMG and radiation overlap in causing DNA damage? What if a newly developed drug induced DNA damage through multiple mechanisms interfere with the DNA repair, indirectly amplify reactive oxygen species, and suppresses IAP expression? Furthermore, if these events occur simultaneously or sequentially, how can we explain this phenomenon? Perhaps we should call this extended synthetic lethality. A single mechanism of action cannot fully explain the effect of many drugs. So there are some reference. I'd like to provide a table comparing the concept of traditional and extended synthetic lethality. Now let me introduce one recently approved drug as an example. API is dordaviprone. Product name is Modeso. Dordaviprone hyperactivate the mitochondrial CLIP protein, thereby inducing mitochondrial stress and apoptosis. At the same time, it blocks the abnormally expressed dopamine receptor D2, suppressing tumor signaling pathways and further promoting apoptosis. Through these two mechanisms, it selectively targets tumor cells while minimizing effect on normal tissues, and it is effective against aggressive treatment-resistant tumor. As above, I explained four key elements that are crucial in VSPharmTech's drug development process. We have successfully secured the four key elements and have identified the shortcut to apply patients. Furthermore, VSPharmTech received orphan drug designation from the US FDA for our glioblastoma treatment just 6 years after beginning development of VS101 in February 2019. Oncologists refer combination therapy, which use anti-cancer drug with diverse mechanisms of action as the most reliable method for cancer treatment. Ultimately, the extended synthetic lethality strategy is not simply a hypothesis or a claim by some researchers, but rather a core mechanism of combination therapy already being used in clinical practice. Actually, many researchers are focusing on combination therapies to improve anti-cancer outcome. It doesn't matter whether it's synthetic lethality or extended synthetic lethality. The really important thing is what's the important is through the development of these treatments and the therapies, patients can get access to treatment opportunities. Finally, just as dordaviprone received accelerated approval as an apoptosis inducer for glioblastoma treatment, VSPharmTech will also strive to obtain accelerated approval for its apoptosis enhancer, which utilizes a non-toxic synthetic lethality strategy as a treatment for glioblastoma in order to provide treatment opportunities to more glioblastoma patients. Thank you for your time.
So we've got time for like one question, if anybody has a question that they want to ask. Okay. Well, if not, then thank you again, and we'll save some questions for the lunch.